Hello everybody. Welcome to our webinar Wednesday. My name is Monika Burbach. I am product manager for biochemicals at Biozyme with 20 years of experience in molecular biology and biochemistry. We are proud that so many people, customers and curious scientists, workers joined our webinar today. Thank you. The aim of our webinars is to transport new and exciting information about technology and new products to all interested people. Often, marketing material is not enough elaborated to understand all details and the complex contexts that lead to superior performance and better results. Today, we will introduce you the information about cell-free protein expression. I'm happy that Dr. Tatsuo Serikawa from biotechrabbit will tell us more about the rapid translation system. At the end of the session, there will be time for a discussion. Please type your questions into the chat box whenever you like. We will answer them at the end. Thank you in advance for your input. Tatsuo, please start with your interesting talk.
Thank you, Monika. Hello, everyone. Thank you for joining. I'm Tatsuo Serikawa, a scientist at biotechrabbit. I'd like to thank Biozyme for giving us the opportunity to share about our cell-free translation system, RTS. First of all, please let me introduce you our company, biotechrabbit. Biotechrabbit was founded in 2011, and the company is located in Berlin, Germany. Currently, about 30 people that are scientists, experienced managers, and business developers working globally. Biotechrabbit obtains ISO 9001 and 13485 certifications. Our company focuses on contract manufacturing for diagnosis and life sciences, especially protein fermentation and development of new proteins, reagents, assays, and kits. They can be produced as bulk and custom formulations, and we offer individual labeling and packaging and lyophilization. Regarding antibody services, we offer mono and polyclonal antibody generation and their large-scale production. Further, our focus relies on development of life science products. Here we offer molecular biology enzymes such as enzymes for PCR, reverse transcription, quantitative real-time PCR, and others. In addition, nucleic acid purification systems and the field of protein research and cell-free protein synthesis are also our focus. For protein research, TRIS-NTA is a very useful tool. The remarkable feature is its high affinity to histidine residues. As can be seen in the right figure, 3 NTA group binds histidine residues. The affinity is about 10,000-fold higher than mono NTA. Using this advantage, TRIS-NTA can be applied for protein detection, purification, and further applications. biotechrabbit holds the global license to manufacture and sell TRIS-NTA. Let's move on to the main topic of today, Rapid Translation System, shortly RTS. RTS can be used in a variety of applications, such as structure and functional protein study and protein interaction studies. I'm going to introduce you the technical background, how to express a protein, comparison between in vivo and in vitro expression, and the core technologies of RTS products, and our product lines that cover both prokaryotic and eukaryotic protein expression. Great advantages of the in vitro expression over in vivo expression are to be able to synthesize easily and rapidly functionally active proteins of interest, and to be able to synthesize proteins that are difficult to express in traditional in vivo system, such as cytotoxic proteins. In addition, in vitro translation is an open system, meaning that artificial amino acid can be easily incorporated into the target proteins. And for each type of target, the reaction condition can be easily optimized. Here you can see the workflows of in vitro and in vivo system. In vitro system does not require time-consuming steps of transformation and cell cultivation. This advantage enables the swift synthesis of target proteins within 2 to 24 hours, whereas traditional cell-based system usually takes more than 2 days. Another great advantage of the in vitro system is that the user needs only a temperature control device, such as thermomixer and pipettes. On the other hand, for the in vivo system, many laboratory equipment and attention of laboratory biosafety are required. As can be seen in the figure, usually for protein expression, template DNA, enzyme machinery, here T7 RNA polymerase, ribosome, cell brick, and energy source are needed. In vitro translation kit, usually all the required components are prepared, ready to use. For example, the lysate is extracted from the living cells, which contains the RNA polymerase, ribosome, and so on. And amino acid is prepared as a mix. Reaction mix, feeding mix contain NTPs. So for in vitro translation system, you need only your own DNA template that can be as a linear or circular form. Our RTS kit obtains all the required components without nuclei, mitochondria, messenger RNA, and cytoskeleton. So only the target-specific messenger RNA is expressed. That means only your target protein is expressed afterwards. Taken together, using cell-free system, only protein of interest is expressed. Time is saved, and no attention of GMO is needed. Target proteins can be easily labeled for structural studies, and proteins that are difficult to express in the traditional in vivo system, such as cytotoxic proteins, can be synthesized. In vitro translation is an open and adjustable system and is scalable from 20 microgram to 50 milligram for a variety of applications. This enables easy establishment of high throughput approaches. Let's move on to our in vitro transcription translation system, namely rapid translation system. RTS is a tool for accelerated scalable protein production with high yield. The target messenger RNA is generated by T7 RNA polymerase. And the messenger RNA functions as a template for translation reaction. All factors that are necessary for the reaction, such as T7 RNA polymerase, ribosome, tRNA, and others are present in the kit. The RTS enables convenient and efficient cell-free expression of functional proteins using key technologies that are coupled transcription translation reaction, continuous exchange cell-free principle, shortly CECF, and high-yield biochemistry. Cell-free protein synthesis is a very simple and powerful system for protein expression. However, the bottleneck is byproducts that are formed during the translation process and inhibit the reaction. As can be seen using conventional batch system, already 2 or 4 hours, the target protein yield reaches a plateau phase due to the limitation of energy source and increased inhibitory byproduct. So the inhibitory byproducts are shown here with green color that are accumulated here in the reaction room. To solve the bottleneck, we have a unique technology known as CECF. CECF system enables continuous supply of energy source needed for the translation reaction, as well as continuous removing the inhibitory byproducts. Therefore, translation period can be extended up to 24 hours, and that enables further increase of target protein yield. Here you can see the comparison between batch system and CECF system. Using the batch system, already after 2 or 4 hours in a reaction, the target protein yield reaches a plateau phase. On the other hand, CECF system enables continuous exchange of the energy source and the inhibitory byproducts. Furthermore, our glycolysis-based energy regeneration system and enhanced buffer conditions enables further increase of target protein yield up to 10-fold. We offer 3 CECF devices for easy scale-up. There are RT100, 500, and 9000 devices that have 50 microliter, 1 milliliter, and 10 milliliter reaction volumes, respectively. The reactions are run in the reaction chamber that are separated from the feeding chamber by a dialysis membrane. So the gray arrows show the reaction chamber. So here the small room, and the green arrow shows the feeding chamber. Here the big room. So here is the feeding chamber, and red arrow shows the dialysis membrane. RTS reactions are run on a shaking and a heating device, such as Eppendorf thermomixer C, connected an adapter for micro-titer plate. If you don't have such device in your lab, no problem. Other equipment that can shake and control the reaction temperatures can be used for the RTS reaction. Taken together, the batch format has quick and easy and flexible options. For example, this is suitable for protein screening, but the protein yield is limited. On the other hand, CECF format enables protein expression with higher yield. This is therefore suitable for antigen production and functional assays. There are 2 options for template generation, linear and circular template. Linear template system enables a fast template generation by PCR. The linear template has, however, a limited stability and reaction period, resulting in lower yield. Therefore, this is recommended for batch system, whereas in the circular template system, the circular template has higher stability and enables longer reaction time, resulting in higher yield, because the circular template is generated by cloning. The circular template can be used for batch and CECF mode. We offer linear template generation kits and cloning vectors for easy template generations. The linear template is generated through 2-step PCR. Here the first PCR and the second PCR. The target in the first PCR, the target DNA is amplified with gene-specific primers containing the linker that are complementary to the fragment in the second PCR. In the second PCR, regulatory elements and affinity tag sequences are added. This PCR product can be directly used as a template for RTS reactions. Our RTS series covers both prokaryotic and eukaryotic lysate system, E.coli, insect, and wheat germ. Our prokaryotic RTS series is based on E.coli lysate system. RTS E.coli system enables protein expression up to 6 mg per milliliter. We have both batch and CECF modes and a disulfide system for expressing disulfide bond containing proteins. We have 2 types of eukaryotic system that are wheat germ lysate and insect lysate-based system. Using wheat germ system, up to 1 milligram per milliliter protein can be synthesized. We have both batch and CECF systems. The wheat germ system has the highest success rate for a broad type of proteins. Insect kit has lower protein yield up to 50 microgram per milliliter, and the reaction mode is batch system. The great advantage is that the insect system enables protein expression with post-translational modifications, such as glycosylated membrane proteins by translocation of proteins into membrane-based code. Here you can see RTS E. coli high-yield kits with 3 different reaction volumes. The smallest kit is RTS E. coli 100 high-yield that has 50 microliter reaction volume and up to 20 microgram of expression capacity. So this is especially suitable for screening. We have kits for 6, 24, and 96 reaction. The reaction mode is batch system, and both linear and circular template can be used as a template. The next bigger size is RTS 500 E. coli Protomaster high yield with 1 milliliter reaction volume and up to 6 milligram of expression capacity. This kit contains reagent for 5 reactions and CECF mode is used. The recommended template is circular DNA. The biggest size of the RTS E. coli series is RTS 9000 E. coli high yield with 10 milliliter reaction volume and up to 50 milligram of expression capacity. This kit contains reagent for 1 reaction with CECF mode. The recommended template is circular DNA. Next type of E. coli system is RTS E. coli disulfide kit. We offer 2 different reaction volumes, 24 reaction of 50 microliter and 5 reaction of 1 milliliter. The reaction system is CECF mode and circular DNA is recommended template. Using 1 milliliter reaction volume, up to 2.5 milligram of protein can be expressed. The key point of this kit is to enable the expression of functional proteins that contains disulfide bound with increased solubility. So this is an ideal tool for expressing antibody fragments and secreted enzymes. This kit is developed based on chemically activated lysate and disulfide isomerase, and there is no equivalent on the market. Here you can see the comparison of soluble proteins between conventional system and disulfide system. Proteins tested are insoluble when they are expressed by the conventional system. Here you can see. On the other hand, using disulfide system, most of the proteins are expressed with higher solubility and enzymatic activity. Let's move on to our eukaryotic systems. RTS with Germ kit enables eukaryotic expression with higher yield. Eukaryotic proteins are often targeted for high academic and pharmaceutical researches. However, E. coli systems are often not suitable for these target proteins, leading to hardly any or a lot of insoluble product. While these issues can be addressed in RTS E. coli to a certain extent, for example, optimization with chaperone or detergent, the required effort is often a bottleneck, especially for high-throughput screening applications. On the other hand, RTS with Germ kit enables expression of eukaryotic proteins without extra optimization. We offer with Germ kit with 2 different reaction volumes, 24 reaction of 50 microliter and 5 reaction of 1 milliliter. For the smallest size, we offer 2 different reaction systems, batch and CECF mode. For the bigger size, CECF mode is used. Linear or circular template DNA can be used. Using 1 milliliter reaction volume, up to 1 milligram protein can be expressed. The RTS with Germ kit are suitable for small-scale expression, high-throughput screening, and expression of a broad type of proteins. For example, Eukaryotic proteins that can be difficultly expressed in E. coli, such as steroid hormone receptor, can be expressed as a completely soluble protein without optimization. Here you can see SDS page, and the targeted protein here is expressed by E. coli system. Here the target protein is expressed by Germ system. A collaborative project with GeneCopier enabled a comprehensive expression profiles of more than 10,000 human ORF using RTS with Germ kit. This showed 86% success rate within the test samples, and this is quite high. So for example, proteins tested are the transport protein, cytokine protein, protease, kinase, and so on. The last RTS kit that I would like to introduce you today is RTS 100 insect membrane kit. We offer 50 microliter reaction volume with different reaction numbers. The reaction system is batch mode, and either linear or circular DNA is used as a template. The expression is low, however, this kit enables robust expression of a wide range of eukaryotic proteins with post-translational modifications, such as membrane glycoproteins. This kit is developed using natural membranes, and this is the first commercially available kit based on cell lysates derived exclusively from insect cell cultures. Here you can see RTS kit overview that I have introduced you today. We offer 3 types of E. coli high yield system and 2 types of E. coli disulfide system. So this is the smallest size 100, and the next bigger size is 500 and 9,000. And here we have 2 types, 2 reaction size of disulfide system. And here you can see the expected protein yield. For eukaryotic expression, we offer 3 with germ kits and 1 insect membrane kit. So the smallest with germ kit 100, we have batch and CECF mode, and the bigger size CECF mode is used. And for RTS insect membrane kit, batch system is used. The following linear template generation kits and cloning vectors are highly recommended for generating DNA template for each lysate system. Optionally, affinity tag can be added to the target proteins. Here, for example, haystack. Taken together, if you start with protein expression for the first time, please start with our cost-effective RTS 100 E. coli high-yield kit. If you have problems with protein solubility or activity, please start with RTS 100 E. coli high-yield kit and RTS 100 with germ kit for eukaryotic proteins. If you need to express active proteins with disulfide bonds, RTS 100 disulfide kits are recommended. If you need to express milligram amount of active proteins, at first, please optimize the condition in the smallest kit RTS 100. For example, reaction period, temperature, and template concentration, and so on. Then scale up to RTS 500 threes. For further scale up, more than 10 milligram, we offer RTS 9000 E. coli high yield. If you want to screen membrane protein expression in natural membranes and proteins with post-translational modification, please choose RTS 100 insect membrane kit. For RTS kits, we have customers worldwide. If you are interested in our cell-free system, please visit our website www.biotechrabbit.com, or please feel free contact us and our partners Westport and Biozyme. Our team would be happy to support you. With that, I would like to thank you very much for your attention, and we'll be happy to hear your questions.
Yeah. Oh, Okay, thank you, Tatsu.
Thank you too.
It was a great talk. And now, just to say, make it clear, if you need further information codes or something like that, and you are from Germany and Austria, you can contact Biozyme directly. And for our friends of Benelux, please contact Vesberg. And now we are open for the questions. So I see the first one. Does the protein size affect the final concentration and yield of the expressed protein, Tatsu?
Yes, I would say the expression protein yield can be affected by different factors, such as you said, as you asked, size and sequence of target proteins and the sequence of regulatory regions of backbone plasmid. Generally, simple and short proteins can be expressed in high amount with our RTS kits. On the other hand, for longer or more complex proteins, the wheat germ kit and insect kit are preferred. Since these are still biological systems, the colon usage also has to be taken into account. Yeah.
Okay, and I have another question here. Is it possible to use my own expression vector for the RTS system?
Okay, yeah. The important point is your vector is under the control of T7 promoter, because all our system use a T7 promoter and T7 RNA polymerase. If you have such type of vector, this should also work in our RTS system. However, for example, in the RTS E.coli system, if the lac replacer is present on the vector, the expression may be suppressed. So in this case, we recommended to add IPTG into the reaction for better performance. We therefore offer cloning vectors for each RTS systems that are designed for effective cell-free protein expression. Yeah.
Okay, so thank you, Tatsu, again.
Thank you.
And I want to wish everybody a wonderful day and hope to see you back to another webinar of our Webinar Wednesday series. Bye, everybody.
Bye-bye.